Okay, I know most of you, but hi everybody. So some of you who know my work won't get much surprise from what I will say, except that you will see. I mean, the surprise will be that I have to jungle between PowerPoint and other application for the movies. So I hope that you won't find it annoying. So I put that title, you know, when you put a title, give me a title for the talk. So you go, oh, blah, 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 5 seconds of thinking, and then you go with the title. So I realize now it's pretty presumptuous. It's pretty almost obnoxious. But think of it as ambitious, let's say. The objective of that talk would be more than just announcing a great breakthrough in cognitive neuroscience, rather just tell you that there is a useful tool that you might use that's in Lyon and for your own research. And so, of course, it's a way to boost and foster collaborations, which is part of the LabEx spirit. So let's start. Okay, so it all starts with that Wolfsinger result from the end of the 80s. Most of you know about that result, that what he found basically, I mean, if we go very to the big headline, would be that basically when two neurons watch part of the same object in the visual cortex, they will tend to fire synchronously. So that creates synchronous neural assemblies, oscillating around 40 hertz, prom, 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 that was the idea, and that would be the basis of perception. So that was a big thing, as you know, at the end of the 80s, early 90s. Then, of course, there was some qualification, some adjustment to that hypothesis. But anyway, it was a very, very strong boost of energy for the field. And it led people like Olivier here in Lyon to look for the existence of such synchronous neural assemblies mediating perception in humans. With very, straightforward idea to look in scalp EEG for increase of energy in that famous gamma band around 40 hertz. So when someone sees an object, when there is visual perception in a human being, is there an increase of power in the scalp EEG around 40 hertz? That would be a good indication of the formation of synchronous cell assemblies in that gamma band. And indeed, they found such increase of gamma band energy, gamma band meaning around 40 hertz and so. And as you can see, I mean, this line of papers in general science which are well cited. Okay, and then slightly later, at the end of the 90s, we thought, I was in Paris, and I thought, well, we have to look for the sources of this gamma band response. We have to be more precise spatially. We want to know where it comes from in the cortex. So the only way to do that was to go to more precise EEG recordings, more precise than scalp EEG, and which is possible with a collaboration with epileptic patients who have electrodes inside their brain, so intra-cranial electrodes. There, the spatial resolution of those recordings is much higher than scalp EEG, and you can actually get some knowledge about the sources of the gamma band response during visual perception. So that's what we did. So this is just to show you the technique that is used mostly in France, but in centers like Lyon and Grenoble to record intracerebral activity in epileptic patients. I show here, you see those depth electrodes, those linear electrode arrays. They will record brain activity over a number of contacts. I don't know if I have that little mouse here. You can see the contact sites with the cortex, with the brain tissue, which are one every 3 millimeters, roughly. So you get a pretty good sampling along a linear array in the brain. And electrodes like this will be positioned, so of course in regions depending on the type of epilepsy of the patient, but there will be typically 10 to 15 depth electrodes such as this one. So you can record brain activity in an epileptic brain in about 100 to 200 sites. The spatial resolution of this recording is around a couple of millimeters. So think of it as something somewhat equivalent to the spatial resolution of fMRI. And of course, the temporal resolution is as good as EEG, so millisecond temporal resolution. So we did that. And so, okay, so just a couple of words to say that, I mean, it's good to know for you, especially if you want to move to intracranial recordings. for those of you who don't, that patients usually they typically have their electrodes in the brain for more than 10 days. And there's plenty of space of opportunities to work with them, have them participate in cognitive tasks. So it's really a good way to get a good look at the brain with high spatial and temporal resolution during cognition. Okay. And so if you do that, and that's what we did, if you look at the signal that you can record from such electrodes, here you have an electrode with an example of an electrode which is located, which records from the fusiform gyrus. You can see it up there, right? The fusiform gyrus would be here. You get, when you show pictures, like this one, so we showed faces, tuck, 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 that's what you get, basically. That's what you get. So you will get several components. Some of them are very well known. So the first one, the most prominent one being the event-related potential. And that's, I mean, the study of event-related potentials in intracranial recordings in epileptic patients had been used for 30 years or even more. So in humans, collaboration between clinical and research groups. So that's not new. That's the old wave, let's say, of human intracranial electrophysiology. But then you get also other components. So you get the alpha rhythm that you can see here that typically would disappear when you show the stimulus, visual stimulus. So you get this suppression of alpha activity that also was well studied and well known. You get also another component which is the beta rebound. So see this oscillatory activity which is slightly faster, between 15 and 30 hertz, which usually comes around half a second later after the stimulus is gone. And then in the middle, you get that tiny, tiny activity which corresponds to the gamma band. So that would be the gamma band response. So that's what we found when we put electrodes into the brain of those epileptic patients and look specifically for gamma. So the news at that time, by the end of the 90s, was that yes, we could find a gamma band response in intracranial recordings in humans, and that this gamma band response was well matched the frequencies that you could record from the scalp, and also was modulated by perception and so on. And of course, at that time, the advantage of the spatial resolution of intracranial recordings was that we would get a good view at the mosaic of the gamma band responses with their extent and localization in the brain. So it was not big response that you could record on the scalp, but several responses well organized in space and time. Okay. And actually, you can use this sort of tools, time frequency representation, and what you can see is a clear increase of energy after the stimulus presentation that covers a very broad frequency band between 40 and 150 hertz. So that was a surprise for us because we expected something around 40 hertz, and what we found there was something which was much broader in terms of frequency. Okay. So which is funny and somewhat ironic. So that was, you see this how ironic from the beginning, from the first slide on, is that we got Wolfsinger visiting the lab last year, and Wolfsinger said, "Oh, that's great. I mean, that's interesting, but that's certainly not my stuff." So that's kind of funny, because if you see how the motivation went, so the whole motivation to study high frequencies in human brains came from the work of Wolfsinger, so with those very nice oscillations, synchronous oscillations around 40 hertz. Then there was the work from Olivier and others, so finding the same, gamma band responses at the scalp level. And then we found an increase of energy during visual perception in the gamma band also, but the frequency band was so broad, so much broader, that it was not possible to call it an oscillation, and that it was certainly a different phenomenon. So although the whole inspiration came from the work of Wolfsinger, we came up with something completely different, with, of course, that question, what is it now? So now we tend to not call it high-frequency oscillations or even gamma oscillation, although, as I say, friends still call it gamma, but we tend to call this broadband activity high-frequency neural activity, or HFA. And I refer to that paper that we wrote with some representatives of the main groups who've been working at the intracranial level in the last 15 years and contributing to our knowledge about high-frequency activity at that level of recordings. And we decided to call it high-frequency activity there. Okay. So what you have to know is that high frequency activity, HFI, is really worth your time. It's really worth your time. And the main reason for that is that it's extremely tax sensitive and stimulus specific. So here I show you an example. So in that study, so we record from the physiforce area, gyrus, and you know that there is a region which is specific to faces. And here it's a simple experiment where we basically show, it's a visual localizer, so we show different types of images. As you can see, landscapes, words, animals, objects. And here I show you the response to faces, houses, and then scrambled pictures. And what you can see here, what I plot is simply the variation of energy in the gamma band, in that broad gamma band, 40 to 150 hertz, to that the level of energy at those high frequencies, as a function of time, when I show faces. So you see the increase, which is pretty considerable. And then when you show other types of stimuli, houses, and then scrambled, almost nothing. And another way to visualize the selectivity of the fusiform face area here is to visualize the increase of energy, so as a function of time again, and for every picture that was shown during the experiment. So each line is a picture, it's a single trial. And what you can see is that there is a very clear increase of high frequency activity for every phase that was shown, with a very reproducible timing, and that's almost nowhere else for none of the other categories, except maybe animals. But here you can see that in some pictures of animals, then you get some faces. So it sort of makes sense. So very, very selective. And actually, then you think, okay, check time, okay. Well, that's interesting because we have a marker that is extremely selective. So that can provide functional maps of the human brain. But then wait. I mean, other people are working on functional maps of the human brain, of course. I mean, those people do fMRI. So fMRI has been providing very nice functional maps of the human brain as well. So you get two maps. So what do you do with two maps? So that's a problem. That's a problem, unless you have to solve that problem. And the only way to solve that problem is to find that there is a nice correspondence between the two maps. And that happens to be the case, fortunately. So that's a study we did a couple of years ago, just having the same patient perform the same task. It was a language task with intracranial recordings, and then remove the electrodes. So there we look for gamma, for high frequency activity, and then they go in the magnet and we do the task in fMRI. And we found very nice spatial correspondence between sites with increase of high frequency activity and sites with bold increase too, when you compare two conditions, semantic, phonological, and so on. But now, I mean, other work has been done more precisely, actually. If you think of animal recordings, the beautiful evidence for that fact from logbook Tetis, from the group of Kim also, with simultaneous recordings in animals, bold and electrophysiological. And then also more recently, the work by Carmichael and colleagues in London, where they do simultaneous intracranial EEG recordings with fMRI. So that's the perfect way to compare the two, right? And there is actually, I think there is one group in the world doing that. We're going to start pretty soon with Carolina Chumas and Philippe Rivelin. But they found an excellent, actually almost perfect correlation between activity at high frequencies, above 70 Hertz to 150 and so, and the ball signal. That's during movement execution. So you can think, if you want to think simple, you can think as the high frequency activity as the best electrophysiological correlate of the ball signal, which means that you can think of high frequency activity and the sort of maps that we provide as fMRI plus time resolved fMRI in a way. Okay, and actually there might be a more general explanation and reason for that good correspondence. And the trend now about the functional meaning of this gamma activity, of this high frequency activity, what is it? What does that mean at the neural level? Is, I think, well summarized in that study by that's Nature Reviews Neuroscience 2012, so June's pretty recent. So that's Buzaki, Kor, I mean, usually they know what they do, Anastasiou, and basically the idea, which is validated by a couple of experimental work, is that what you get in those local field potentials recordings that you get with intracranial recordings is a contamination by spiking activity. So this high frequency activity that you get, this huge high frequency activity, would actually be the trace of spikes. So that means that if you count, if you measure the energy in that broad frequency range between 40 and 150 hertz, what you will get is basically an estimate of the number of spikes that were produced by the neural population that you record from. So that's spike activity at the population level. So then it's not surprising to find that it's so tightly linked to cognition, tightly linked to stimulus, to the sort of stimulus that you show, and so on, and tightly linked to the Bolt signal eventually. Okay, so that's good news. Actually, they said, well, I mean, first, everything that you record just an artifact, just contamination by spikes. But then, I mean, well, think. I mean, that's beautiful. It's good news. It's good news. And you get a good marker. So this is the first marker that you get in the human brain of spiking activity at the population level. Okay, I will skip that one because I have another one. So basically, once again, to make the story short, you record here, 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 and then in one of those places you have neurons processing information. They process information. When they process information, of course, they generate spikes. I mean, they send information to the rest of the brain, or even just locally. Then you will have an increase of high-frequency activity. Okay, that's just straightforward. And then, of course, it goes the other way around, which means that when a neural population will stop processing information and stop working in a way, they will stop sending information, stop generating spikes, or have a reduced number of spikes, and then the high-frequency activity will go down. So you can see increase or decrease of neural activity just by monitoring high-frequency activity with those intracranial recordings. So that's pretty new. I mean, that's why I said the new wave of human electrophysiology, because 15 years ago, there was nothing about that. I mean, the only thing that was known about high-frequency activity was basically what was being done by Olivier and So in humans. And I mean, of course, you have all the animal works, but in humans, it was basically just the gamma band response in humans. And at the intracranial level, nothing. So it's really a new wave, and more groups are working that way. So, and once again, it's funny. I mean, we are looking for 40 hertz synchrony, and then we got something that, I mean, okay. I mean, you need to be cocky at some point. Okay, so I would call it the first real-time marker of local neural processing in humans. Okay, let's be cocky. And then I take questions. Okay, so what can you do with that? I mean, of course, many things. You know, the patients are just across the street. So there's plenty of space for experiments. Just the future is wide open. So what can we do? I'll show you a couple of examples. So that means I have, what, 10 to 15 minutes?
You got into 5,000.
Okay. Okay, cool. So first example. You know, when you read a text, you read a book, something weird happens. Is that you look at things, you look at words, so it's a visual input, and then you get a sound in your head. It's beautiful. I mean, I guess it's the same for you. That's what happens for me. Maybe there are different kind of people. But you look at a word, look at here, word. Okay, you see a word, you see those white lines, and then a sound in your head. It's beautiful. How come? I mean, what's going on? So we had that simple question. We said, okay, let's look at what happens in a human brain, just reading. There seems to be an auditory component. It's actually a voice component. So maybe if we look in the auditory cortex, you know, there are, I mean, first you have the primary auditory cortex, but you have also voice selective areas. So maybe we can record some activity there. And that's what we did. So I don't want to go into all the details of the experiment, but basically we are showing words, and they were forming a story. Some words had to be attended, and other words that you would see in red had to be ignored. And the bottom line is that the attended words generated a very nice high-frequency response in the word form area, which was expected, but also in the primary auditory cortex, and also in the voice selective areas. So indeed, just by showing a visual stimulus, you can activate the auditory cortex. And what's funny is that the two activities, they don't happen at the same time. So it's not a pure audiovisual sensation. It's like one thing and another thing. Plus the latency between the two, it can differ. It can vary. It can vary from one word to another one. For some words, it takes longer to create the auditory component. So that's something, really, if you want to study that kind of questions, you really need excellent spatial resolution, because you need to be able to separate the fusiform gyrus and the auditory cortex. And in the auditory cortex, you need to be able to separate the primary auditory cortex and the voice selective areas, which is more associative. But you also need excellent temporal resolution, right? Because otherwise, obviously, with fMRI, there is no way to find such phenomenon, so latency difference. Okay, so that's just one example of what you can do with that index. And of course, you need a good measure of neural activity at the population level. Okay, so I want to show you another example that we've been investigating a lot, which is visual search. And basically, you know that when you search for an object, sometimes it's easy when there is a big difference between the target and the distractors, and sometimes it's more difficult when they all share the same features. Okay, so what's interesting is that then you can look for activity, neural activity that lasts during the entire search. So when the search is fast, then the activity will just start and then die down pretty early, and then when the search takes longer, then the activity will last longer. Okay, so we are looking for such activities, for regions with an activity that really lasts during the entire search. So that's where I have to be. It becomes technical. Okay, wow, beautiful. So you can create those movies. You just basically accumulate high frequency activity, high frequency measure across several patients. You can summarize. I mean, ask me questions. I go into the details of how you make those movies. And what you can see is as a function of time. So you see the stimulus coming on at 0. Come on, yeah, okay, at 0, and then okay, it flows. So that's me. So it's slightly slower than real time. And then in red, you see regions that start generating high frequency activity. Okay, so you can see a beautiful and expected activation of the visual cortex. And then you get also an extension of this activity, a propagation of this activity, an invasion of the frontal lobe, and in regions that constitute the dorsal attention network. And what you can see is that if you compare two conditions, you compare a fast search, that would be at the top, and you compare a slower search, so it takes longer. You can see that in some regions, the activity will be sustained longer when the search takes longer. So those regions are really interesting for us because they are the regions that really participate into the search process. So that's something that you can learn from having the time dimension. And I talk about the blue ones right after. Okay. So a way to actually display the activity, the high frequency activity, is to sort the trials according to reaction time. So here I show you the activity measured in 4 different regions. And simply, so those are all the trials, all the searches, and this is the reaction time. So sometimes it's fast and sometimes it takes longer. And what's interesting is that then you use time to really distinguish functions, because you see the fusiform gyrus will basically just react initially to just the onset of the stimulus. Okay, there is a stimulus there, not much. So this region is certainly not involved in the search. Then you see the motor cortex. Activity in the motor cortex will be centered on the motor response, so expected. And then you go to other regions like the frontal eye field or the dorsolateral prefrontal cortex. And then what do you see? You see an activity that will last during the entire search. So when the search takes longer, then the activity takes longer. So then here you use time to really say that those regions are really associated to the search. And then you can go into more subtle details because then you can compare the fine dynamics of those responses and look. So here in the dorsolateral prefrontal cortex, activity just dies down slightly before the motor response being I found the target. And if you go to the frontal eye field, then it takes a little bit longer. Well, it lasts almost to the end. So you can say that those two regions perform different functions because you can separate those functions based on timing. So that's something else that you can do in humans, pretty much for the first time. Okay. Here about underactivity suppression. So I show you there were some nice blue spots. Okay, those blue spots correspond to suppression of high frequency activity during the search. So at first we thought, well, what's going on? What's going on? What's going on? Okay, so that means that there was a high frequency activity, some gamma activity just before the stimulus, and then it disappears. What's going on? Okay, and then someone comes with a nice review on the default mode network. He says, okay, look, that's the default mode network, and then we look at our suppressions of activity, and we go, oh man. I mean, there's a pretty cool match between the two. So you see the temporal parietal junction, you see the posterior cingulate cortex, major prefrontal cortex, and so on. I mean, it's the same network. So it's the same network. So it's a way to rediscover the default mode network. That's interesting. It's completely independent of fMRI. And then you use intra-cranial electrophysiology and you rediscover a set of regions which activity is suppressed when attention is directed toward the external world. But, okay, well, great. Okay, you rediscovered the default network 10 years after everybody. That's wonderful. Okay, well, yes, it's wonderful. It's wonderful because you get time again. And what you learn from that, from the timing information, is that the suppression is extremely short. The suppression lasts longer when the search is longer. And actually, you can actually correlate with the reaction time and see that the suppression really ends when the motor response is given. So here is a network that cannot be suppressed longer than necessary. So whenever the task is done, whenever it's finished, then it must go back to normal. So we are dealing here with something that is so vital that you must reactivate it as soon as possible. So any interpretation like daydreaming and so on, I mean, you can just throw it to the trash because it's impossible that you would just stop daydreaming for half a second and then immediately when you press the button, you start daydreaming again. So that's impossible. So the default mode network has something to do. It's another function. Okay, so time tells you that. Now, functional connectivity. Okay. Just giving you an example. Everybody is about networks, networks, networks. We want to know about networks. We want to know about connections between regions. They work together. Okay, well, I mean, they work together. What does that mean that they work together? This one works. It processes information, sends spikes. So high frequency should go up. Then that other one is also working at the same time. So high frequency should go up, but should go up at the same time. So that means that if those two regions work together, then high frequency activity should be correlated in time. I mean, I'm not talking about phase synchrony here, but something much simpler, simply a correlation of amplitude. So go up, go down, go down, go up, go down, go up. So here we have an example. We got it from two regions, one in the medial temporal, actually the middle temporal gyrus here. Okay, involving social cognition, storytelling, and so on. And then another one in the infrafrontal cortex, pretty much in Broca, anterior part of Broca. Very, very strongly semantic region. And then we had that simple experiment. We asked the patient to read sentences. I mean, you may not believe it, but having an experiment on people reading sentences, actually, it's almost impossible with non-invasive methods. Because with EEG or MEG, you get all sorts of artifacts with the eye movements. And then with fMRI, you never capture the fast dynamics, which is paced by ocular exploration. You get 4 saccades per second. How can you study a phenomenon with 4 saccades per second with fMRI? So once again, I mean, this is the first time that you can actually do that. Just study what's going on in a human brain reading a sentence. So it's pretty straightforward to do. And when you do that, you discover that in those two regions, you get, I mean, I will not talk about control conditions, but here, you get an increase of high frequency activity that will last as long as the sentence is read. Whenever the sentence, the patient is done reading the sentence, then stop. Okay, you get that white line indicating reaction time, so it's all sorted by reaction time and stop. So that's a good place to look at if you want to study interaction, because we get a sustained activity in two brain regions during the entire reading process. So, I mean, reading is really something that would need communication within the human brain, right? I mean, you will not, there is no way the middle temporal gyrus is reading just alone. So let's look for correlation. And indeed, we actually found very, very, very strong correlation between the amplitude fluctuations in those two regions. I mean, that's just a trial, but there are tens of those with such high correlations. We're talking about two regions that are separated by 10 centimeters. 10 centimeters. So no possibility of volume conduction whatsoever. So here we're really talking about two regions working together. So the co-modulation of high frequency activity can really reveal functional networks. And that again is pretty new. I mean, from an electrophysiological point of view. Okay, and if I have some time left, I won't talk about Brain TV this time. I just want to show you one movie. So as I showed, there is no need to average. I mean, in electrophysiology, the first time we do is averaging, event-perturbated potential and so on. There is no real need to average because you can see the response in single trials. I mean, now it's the time for experiments where you just have 4 trials, one for each condition, and just compare them. I mean, you can, why not? Why not test that? There is no need for trials. There is no need for events because you don't need to average relative to events. So I'll show you an example where so we found these nicely selective brain regions in the fusiform gyrus, selective to words and consonant strings. I mean, that's well known. There is a word form area. Come on. But that's one, I mean, nice confirmation by electrophysiology here. So you see the gamma band response in pink and blue for everything that looks like a word, and then for everything else, pretty much nothing. And I want to show you that movie and then stop. Where you will hear the gamma in that region when a patient is comparing two scenes, two visual scenes. So it's free viewing. You know, it's the find the difference, spot the difference kind of gamma. And there's text in the picture. So I want you to just pay attention. Hopefully there is, I don't know if there's some sound. Come on. If there is no sound, I'm dead. Well, the signal is shown also on top, but it's less.
You've got 4 minutes.
Okay. Well, let's go. Let's see.
You might want to conclude in that 4 minutes rather than go moving.
Oh, I can hear the. Come on, come on. Okay, I went back. Perfect. Can you hear the sound? That little B, it's the gamma. It's the high frequency activity. So the little white spot, the little white B is the gaze position of that patient. So look, spotting differences. So it's pretty quiet. Can you pop? So it becomes crazy. There is text. There's no event, there's no trial. There's 1 minute, okay. So it's not a revolution. I mean, what you learn here is simply that selectivity in the temporal cortex is preserved during free viewing in humans. But once you know that, once you have that, then you can do interesting things, other experiments. Okay, and so actually you can look for fixations that generated gamma, a strong gamma activity, that's in pink, and you can see that the distribution is very much coincident with the text regions there. Okay, other fixations in green. And just, so once you have that, you can do it in even more real-life situations. So that's the brain TV idea, that you can have gamma in real time as the patient is looking around. And I had an example where you're in the fusiform face area and the person is looking around, and whenever she's looking at faces, then you hear whoa, and when she has that face as a talking face, then it's even stronger, so you can learn like that. And just test hypotheses on a quick basis. And so that's brain TV. And okay, just one last minute. Just one last minute to describe the localizers project. So the patients are right there at the hospital. Here's what we'll do. For every patient, we'll do this little operation. We'll take the brain, 1 minute. We'll get it to work, the brain. Memorization, reading, listening, looking at different visual categories and so on. So we'll perform a little localizer, short localizer for different kind of functions. We'll analyze the data, 3 minutes, and then after 10 minutes, you have a brain, and you have a number of functional maps for all the main cognitive functions. So we'll have that for every patient, which means that whenever you're interested in a specific function in human, you can come to us and say, okay, I'd like to have a patient who has an electrode in the word form area and an electrode in the dorsolateral prefrontal cortex, and why not Broca? I don't know. But the semantic part of Broca, not the phonological part of Broca. Okay. Then you say, oh, we have one because we have run the localizers. So we tell you, okay, there is a patient there. So what experiment do you want to do? Oh, that's very smart. Okay, let's do it. So we'll have that kind of functional map for every patient now on for all the duration of the cortex LabEx. So feel free to, I mean, I think that can be really useful for the entire community here. And that's it. And I encourage you to, if you want to know more movies, see more movies. I mean, the movies that I couldn't show on braintv.org. Thanks. The questions. Can we maybe stop the little bee? I'll do that.
So, with these intracortical electrodes, do you have the possibility of recording from different layers of the cortex? I mean, your electrodes are going right through the cortex. Can you record from different layers?
So we don't know exactly. Yeah.
The gamma is located primarily in the upper layers, and it's a feed-forward activity, and the lower layers are the alpha. Do you think that's because you're summing up the activity across all the layers? You get this very broad spectrum of frequencies?
That's a possibility. What I know is that every electrode will record from a few cubic millimeters of brain tissue. So of course that would include all the layers. So we have no way to know what layer contributes most to what we see.
So you're not using something like Schrodinger's electrodes, which would give you that kind of information?
Okay, the next step, and that's part actually of the IHU, is that we're going to purchase, I think it's almost done now, all the BlackRock systems to do single unit recordings in those patients. So then we have much more precise recordings, so unit activity, and we might be able to reconstruct the layers in which those electrodes are located. But that's really work in progress. I don't know any group, maybe Algren is doing that, being able to actually know from which layers the recording is come from.
Any other questions?
So you spoke about co-amplification or co-deactivation of gamma.
Right.
But I'm sure that you look at phase synchrony, don't you?
Well, actually, okay, phase synchrony in that case doesn't make much sense because you have an activity, as I showed, that is so broadband. So since the frequency band is between 40 to 150 hertz, you cannot even define a phase because you don't have oscillations. And since you cannot define a phase, you cannot define phase synchrony. So the only thing you can do is look for amplitude correlation. Phase synchrony doesn't make much sense there. You could do it, and we've done it, in the beta range or in the alpha range, of course. But for alpha, no. Actually, I must qualify what I've said, because in some brain regions, especially regions with just early sensory regions, and V1, V4 would be such regions, we get a much narrow frequency band which is much more narrow. So we would get the sort of narrowband activity that Pascal Fries would get, for instance. We get something around 70 hertz. And then you get something like an oscillation and you can look for phase synchrony. Okay, so it's not like everything that Pascal Fries have been doing, for instance, is wrong there. It's just that it might be valid in some brain regions, not in other. As you move to more associative brain regions where, let's say, receptive fields are larger, for instance, I mean, it's less organized, then the frequency band seems to be broader, and you cannot use phase synchrony anymore. Where is it? Where's Peter? Pierre.
When you show this gamma activity related to the reading in your final film and in the language processing, we've discussed this recently. This notion that the microsaccades are possibly contributing to the
right.
So, and even in your scanning of the of this image. So how do you have some method to control for that? I'm sure.
Yeah. Well, we're talking intracranial here. It's not that any intracranial recording will be completely free of artifacts from eye movements. You know, I mean, Peter is referring to the fact that whenever you move your eyes, the eye muscles will generate a spike. Electromyographic activity that generates some activity, some energy in the gamma band. So at the scalp level, there is some contamination by those artifacts. That's for sure. At the intracranial level, it's pretty much clean, except in very, very specific brain regions. And we've shown that in the temporal pole, because you're really touching the eye muscle there, then you get some artifacts. But elsewhere, it's pretty clean. And one reason for that is that we work with bipolar recordings, okay, and plus the spatial resolution of the electrodes. But as you can see, I mean, okay, remember that graph where I showed the different dynamics in those four during the visual search? Okay, you have the three different gyros, then you have the DLPFC responding completely differently. Of course, if it was an artifact, you would have the same dynamics everywhere. Contamination, you can have, okay. Same during the movie where you see this comparison between the two pictures. I mean, there would be gamma everywhere because the eyes are constantly moving. While here you see it's specific to relators. So the signal is pretty clean. And of course, I didn't have the time to say that, but of course you get some epilepsy from activity because those brains are epileptics. So you must be careful. So come to me, to people working with epileptic patients, and I mean there are ways to get around that and to make sure that the recordings that you work with are clean.
Do you have a patient, children, and how is high frequency activity in this patient comparing to the adults?
If we get high frequency activity in kids, yes, we do. I mean, we've been recording for a year or so at the HFME with kids. But it's true, I mean, we got some nice responses in visual areas, high gamma, no problem. But it's true that usually the signals were not as convincing as here. So we don't know why. Maybe, I don't know. That's something to be investigated. But definitely in visual areas, for instance, can generate beautiful gamma high frequency, no problem. Yes, also in kids.
What is the strength of correlation between the ball signal and the high frequency gamma activity?
Yeah.
Because the first one was thought to be related to synaptic activity rather than spike activity.
Yeah.
So and you said that the high frequency gamma activity was related to spike. So
okay.
What is the answer?
There was a very nice talk at the Jacques Cartier Entretien 2 days ago from Carmichael, David Carmichael. So David Carmichael is the one, because there was only one, in London, in the world actually, so recording simultaneously intracranial EEG and the fMRI and the ball signal. And what he showed us the correlation between the electrophysiological activity and the ball signal as a function of frequency. And he said that the correlation goes up to 0.8 or 0.9 when you go above 40 hertz or 50 hertz. And what he said, and I really clearly remember that, is that the correlation between the ball signal and high frequency activity was even better during a movement task. That was in the motor cortex, even better than between the ball signal and the paradigm, the experimental design. I mean the regression function, like act, stop, act, stop. So the correlation was better with the gamma than with the paradigm in the motor cortex for a movement execution paradigm, which is, I think, pretty strong and pretty illustrative.
Any other questions? I found your result with silent reading interesting. 700 millisecond delay between the visual and the auditory is kind of huge.
Yes.
People think silent reading evolved very recently. So if you were to ask your people to read aloud, my bet is you wouldn't get that activation. You'd have to cut back the feedback. You wouldn't have to hear their own voice. But they wouldn't be having that effect that you get, that magical inner voice wouldn't happen when you read aloud.
Why? It's possible, especially since, and actually that's the patient we have right now in Grenoble, has an electrode in the voice, in temporal voice area. And we could show very clearly and could observe again and again that there is a very strong reaction to voices, but external voices. And when she speaks, there is no response because there is this, this phenomenon of just inhibition. So that's a very good point.
Read aloud and see if you don't have that response.
Yeah, true. That would be a good point. Actually, we got some recordings. We asked her to read. We had an electrode in the word farm area, and we asked her to learn katakanas, Japanese characters. And she had at some point to, after learning them, she had to tell them, to read them aloud. So we have the recordings, so we can look at that for sure.
Is there a burning question? Okay, thank you very much.
Thank you.